Our first speaker is Niklas Rindtorff, who is from Lab.Bio, which Lab.Bio is basically aiming to make computational biology easier for us all. Niklas is the founder of Convexivity Labs and the president of Open Lab Association. Thank you so much. Here he is.
Thank you. Hi, good morning, everyone. I'm Niklas, founder of Convexivity Labs and the Lab.Bio project, where we're building Lab.Bio. And the goal for Lab.Bio is to make computational drug development as accessible as possible, which I believe is particularly relevant for decentralized science. And we're going to walk through why that is the case in just a moment. And I'm here today to tell you a bit about what have we been up to and what I hope everybody can take away as what you can do within your DAO and the projects that you're funding. So I think 2 to 3 years into decentralized science, it's fair to say that the killer app for DeSci up to this point has been funding, and a particular ambitious type of funding. The idea that you can fund a large number of diverse projects very early, at high risk, and with very, very small relative amounts of funding. And how small these relative amounts of funding are and what the nature of these projects are is something that I'm going to take a look at just briefly. So these are 3 somewhat randomly selected projects. I think it becomes pretty clear that I'm a VitaDAO fan, that were recently funded. And when you look at the price point that has gotten funded, usually a ticket is about $300,000. And the nature of the project is always at the point where a target has been identified, some form of drug target, be that an ECM target like Matrix Bio, an autophagy target, or a senolytic target. Every time you know a protein within the human cell that is associated with some disease, and the purpose of the project is to A, validate that that is actually a legit target, and then B, find some kind of molecule that binds tightly to that target in order to actually treat the condition in some meaningful way. And I think at this point it's fair to say that online communities have been pretty effective at picking targets, picking projects that go after promising targets, but I think what's still not absolutely clear is how good are we at actually taking these targets that have been selected in a group and then turning them into actual candidate molecules that can turn into medicines. And I think to just establish a bit of shared truth here for this room, it might be useful to go through what are the actual steps that happen after I know that I want to go after a drug target. Okay, so this is all preclinical development. And you can separate it into 3 stages. And the first one, it's hit identification. So basically, you search through chemical space. Those could be small molecules, those could be protein binders, those could be conformations of the enzyme that you're trying to engineer. And you try to identify the configuration that has the desirable property, which in the beginning is most of the time just, does it bind? Does it bind tightly? Does it inhibit the activity of the enzyme I'm trying to stop? Or does it bind tightly to the surface marker of the cell that I wanted to remove? And then in the second phase, it's the hit to lead phase, you actually verify that whatever came out of your first screen is actually legit through something that is called orthogonal assay. So you basically try to prove the same thing through different means. And then you enter this third stage, which is lead optimization, where you take something that worked in the beginning and you iteratively make small changes to turn it into actually something that you would dare to put into a human. Now let's look up how much these different phases usually cost when you look into traditional biotech companies. And it's a pretty good source for this, for open source models at Bio that do a really good job at aggregating a lot of information on the sort of likelihoods of success and costs of individual project stages in biotech. And when we look at these costs, they're nothing like the type of money that we're usually spending in DeSci projects. So even going through the hit identification in a rigorous and professional manner usually costs you about $2 million based on this model. And obviously we have to take this all with a grain of salt because these data are somewhat selected. If we go further, the cost just keeps compounding. We're looking at $5 million to go to hit to lead, $10 million plus to go through lead optimization. That's all before you're even in the clinic, and your probability of FDA approval starts rising, but it's still not at the level where people would be super comfortable. It is venture after all. But I think one number that really sticks out to me is this $2 million number. So basically, the benchmark for industry to go from finding a target to actually having a molecule that binds to that target is at 2 million, yet the amount of money that we're putting to work in decentralized science is about 1/10 of that. And if we believe to have somewhat equivalent levels of return, we need to really get a bit creative and think about how can we get about the same quality for a fraction of the amount of funding that we give these teams. Now, the upshot is, I think we have a solution, and the solution lies in the primary driver of cost, which is actually you spend a lot of this money in the laboratory paying for physical experiments and paying for the reagents that are required to do them. And this is something that I think we can dive in a bit more, but first let's establish a bit of nomenclature so we can go and I'll follow through the coming slides. So the primary question that a lot of these currently funded IP NFT projects have, and what a lot of the money is being spent on, is which molecule binds to that target. The scientists applied for funding, they had a particular target in mind, they'd already done years of work to find a target, and they now want to use the funding from VitaDAO and other BioDAOs to actually go and find a drug that hits that target. Most of the money is being spent to answer that question, and the space is massive, right? You have 30 billion small molecules that you can choose from, for example, and you really need to narrow it down really quickly to get to about 10 lead-like molecules that you might want to follow up. Now, the good news is, and that's what I'm really hopeful, is that we're in 2024 and things are changing quite rapidly in our ability to model and avoid going to the lab. So this is from a recent paper that probably some of you have seen. This is from the AlphaFold 3 paper, which is now advanced to the stage where we're not only able to fold proteins, I think this is what most people are familiar with AlphaFold, but we're now at the level where we can also model a protein in complex with a small molecule inhibitor, which is still the primary modality to hit a drug target today. So we're basically now at a point where we're able to outstrip all the up to this point, conventional methods of trying to simulate how a small molecule binds to a target, and there are also open source versions around the corner of these algorithms. But simulating how a small molecule binds to a target is just half of the way. You can do this at scale, and you can do this a lot, but actually what you're really interested in is how tightly does it bind? What is the intensity of the binding affinity? And there are also some pretty exciting news coming up here, and this is from Gregory Ross, who is at Schrödinger at the time that this paper was published, who's now at Isomorphic Labs, the DeepMind spin-out. And basically, what they did here is they had a simulation product that they call FEP+. And FEP+ is able to take one of these structures and then simulate how tight is this interaction. And what they did was they looked into historic data where people ran high-throughput assays and low-throughput, sort of very sensitive assays of binding affinity. And in particular, they looked for cases where they had two teams that were not working together test the same target with the same molecules. And then they looked at, well, what's the error? What's the deviation that they get when two teams run the same experiment, basically? And that's what you can see in purple here. This is the root mean squared error. And you can see this is about 1 kilocalorie per mole, which is just the unit to measure binding affinity. When you now look at the accuracy, or in this case, the error that this FEP+ simulation suite is giving you relative to some of these experimental values, the errors are somewhat overlapping. The distributions of errors are about the same. This means we're now, I think, in this extremely exciting time where you can not only predict the structure of a small molecule binding to your target, you can actually measure the binding affinity or simulate the binding affinity at sort of the level of accuracy that you would get out of an experiment. This is pretty exciting because now it actually opens up an avenue how with 1/10 of the money, you can potentially actually get the same amount of results than if you had run a brute force sort of conventional high throughput experiment. So I think it's pretty clear at this point, and especially for people that are doing this like as a full-time job, is that you want to do some kind of simulation before you go into an in vitro experiment. Now, at that point, you still have a problem, which is you have this massive chemical space that I said in the beginning, like these 30 billion molecules. What molecules are you going to select? What molecules are you going to test? Now, while the in vitro experiment costs you about $500 a pop to test one molecule, the simulator still costs you about 50 cents to $5, depending on the amount of GPU compute that you need to put in. Just for perspective, these free energy simulations here, they run for about 5 hours on an A100 GPU. This is no joke. Like an AlphaFold 3 prediction, you can get that in 5 minutes. But getting those simulation results, that's 5 hours of compute. So you want to be sure that what you're simulating is the real deal or something that at least has a fair shot at being useful. So you need to be even smarter than just using a simulator instead of going to the lab. You need to constrain your chemical space. And this is where generative models come in. So not only have we made a lot of progress in simulators that are getting to the accuracy of in vitro experiments, we've also made a ton of progress in generative models that help us basically generate small molecules that have high likelihood of passing through the simulator's tests. And we can basically tune these generators to produce small molecules similar to the way that ChatGPT would produce sentences. These generators produce small molecule structures, and we can condition them based on the target structure to bind against and the size of the binder that we want to look at. And we can do this for both proteins, so designing antibodies or small protein binders, and we can also do this for small molecules. Now, this is pretty complex. And something that we've been doing over the last couple of months is bringing this all together into one integrated platform, where you can basically define the target, you can define the number of binders you want to generate, the type of length that you want to look at, and what we do under the hood is we run a generator and then we run a simulator to verify and give you an estimate of how good your potential generated molecules are. You can then explore the results. So this is, you go through hundreds potentially of generated molecules. You can see what is the stability of these molecules that you generated, what is the binding affinity, and so on. You can manage all your results. They're tracked through an internal IPFS. They're secure with us, but if you want to share them within your DAO or within your community, or you want to integrate into other Web3 mechanisms, you're absolutely able to do that. And I think that's something that is extremely interesting, especially when we think about Catalyst and making it even more accessible to a broad audience to mint intellectual property claims. Then I think that this being able to track your work and under the hood having a wallet that actually tracks everything you do on our platform is a great plus. Now, I want to take you actually through an example, and if I had my laptop here, I would do it live, but I unfortunately just have this video here, and maybe someone in the back can help me play this video. Oh, okay, maybe not. Okay, so, nope, that's not it. Well, I guess we'll just have to do this to a Q&A session, or we meet after the talk and I just walk you through the platform. The platform itself, you can already log in. You go to Lab.Bio, it's pretty straightforward. You can sign in using your MetaMask, or you can sign in using your Google account, and you can currently design protein binders based on the functionality that I just outlined. Now, this all sounds fun, but does it actually work? Like, can we use this? And there's no better way to demonstrate it than just by dogfooding it yourself. So we partnered up early this year with Lab.Bio, a company based out of Switzerland, that actually runs a wet lab, so we can actually test the proteins that we generated. And we picked a drug target for senolytic CAR T cells. So these are CAR T cells that target cells that have gone senescent, which is one of the key mechanisms of aging. And the idea is that if we design a protein that binds tightly to a surface marker of these senescent cells, that we can remove these cells by clicking that protein that we identify to a CAR T cell. And we went in and we stamped out about 3,600 binders. We filtered them down through our simulator, which again was very coarse-grained at that point. And we generated about 6 successful binders. I'm pretty excited about that. Now, this is some of the first data that I'm actually sharing about this. But you can see here 2 of our binders, and I think the first time that we're releasing this publicly, we validated 2 of these binders here. This is the dose response curve. We have a nanomolar range of binding. This is a completely generated protein binder that is not overlapping with any existing binders to this target. And actually, just a couple of weeks afterwards, it was exciting to see that there was another independent group in academia validating this target, again, through a second publication here that was just featured in Nature Aging, where we can see that there is quite a strong signal now that this target actually is able to unleash CAR T cells to delay metabolic aging. Now with that, actually I already want to thank you and give you a bit of an overview of the milestones that we have ahead. We're just a couple of weeks away from launching this platform. We're currently improving still some infrastructure stability issues and simplifying the payment mechanism. One thing that's extremely important to us is that this is usage-based payment. Because when you're running a DAO or you have a couple projects that you funded, you don't want to have to pay for seats. You want to just pay when there's actually usage and when we add value, and the teams that you're funding are seeing the value in it. And that's something that we want to make sure. The next thing that's going to come up is we're going to integrate small molecule design as well. So basically right now we currently only support protein design, but 9 out of 10 projects are really focused on small molecules, so it's important that we really get our leg up in that regard as well. And then I think the not too far future is to really drive up our simulation-based validation by putting in big GPU compute and really getting to that level of experimental accuracy. And with that, I want to thank you, and happy to demo the platform after this talk on a laptop. Thank you.